Lots of announcements for this week because I haven't seen you guys in a couple weeks. So this week we will be doing experiment 20 part C in lab. So remember your technician assignment. You're going to this week be keeping track of what enzyme you will use. So make sure you look that up and have the correct enzyme for your pre-lab. And you use the correct one for the experiment. We'll have our last quiz, quiz 5, this week. And then next week, we're, this is the in-lab part, and then we'll talk about the midterm here. In-lab, as a whole group, we will have our experiment 20 discussion based on the data that you collect this week. Everybody will be completing evaluations. And then the Chem 256A people will be checking out of their drawers. So A only people not taking Chem 256B. And then those taking 256B are going to go on to the library, and we will basically start 256B next week. So this part will take a chunk of time in lab, hour and a half-ish, not more than that based on the library times that are set up. So there will be a chunk of time that Chem 256B people will be doing some stuff in lab as well. And then we will split our separate ways, all right? So big thing in next week, Monday is our midterm. Right now, we will be, a week from now, we will be taking a midterm all together in this room together from 4 to 5. We have, so most of you know, there is a class in here right before our class. So I am going, as soon as they leave, I will have to quick get all the desks set up, get all the exams distributed, and then let you guys in for the exam, and we'll start you at 4 o'clock. So if you could do me a favor next week, wait until I let you know that it's okay to come into the room to get a seat where there is an exam so that I can get everything set up really quick, because we'll have hopefully 10-ish minutes maybe to get the room set up. So in a little bit here, I'm going to go through the practice exam. But for the exam, make sure that you bring pencils, erasers, calculator, ruler. Bring all of these things. You need, I am telling you now, you need all of these things. So make sure that you have them with you, okay? And then if you need paperwork signed for the Academic Success Center, make sure you have me sign it right away. Okay, I need to sign it either today or tomorrow, so make sure you have me sign it. Okay, so we're going to kind of review here a little bit of where we were, and then go over where we're heading with experiment 20. So if you recall from a couple weeks ago when we first looked at experiment 20, the premise of experiment 20 was eventually we wanted to get to precursors of these compounds. So we weren't going to make these exact compounds, but we wanted to get to compounds that we could use to then make these compounds. Okay, so these are what the end result of our work would get to eventually, but these aren't the compounds that you're going to make in the last step of experiment 20. Okay, so just keep in mind that you're making hopefully making the precursor to these guys. Okay. And so, and all of those are in your lab manual. So when you're writing up experiment 20, keep in mind where you wanted to be heading. Well, these are off by weeks here, but first week we did in 20A, we added an alcohol with a Fischer Esterification to our carboxylic acid. And so we made our first ester, our benzoate ester, in the first week of lab. Okay, our first week of experiment 20. Then the second week of experiment 20, we used our acid chloride and pyridine. We made our second ester that week, and so at the end of 2 weeks ago in lab, you should have a diester that is in your drawer. Okay, so now this week we're going to take that diester and we're going to use pig liver esterase or orange peel esterase, depending on your enzyme, and figure out what happens. See what happens when you put it in the enzyme and see if you get to the desired product, okay? And so kind of here again are our desired products. So here are R prime. This is This product is from having methanol in the first step, and then our R over here could have been methyl ethyl propyl. Same thing, R here could have been methyl ethyl propyl, but this is from using propionyl chloride, or sorry, ethanol in the first step, and then this is from using 1-propanol in the first step. Okay. So now we're going to look at first a little bit of general information, as much as information as we cover in organic chemistry about enzymes, and then we'll look at our specific reaction. So enzymes are everywhere in all sorts of living systems all over the place, and they have specific jobs that they do, and they act as catalysts for those jobs to help whatever job they need that needs to be done, help it move along more quickly. And then the goal of the enzyme is then to regenerate itself. So it doesn't want to be consumed, but in the end you want the enzyme back. So enzymes can do all sorts of jobs. We've got transferases, hydrolases, lysases, isomerases, lipases. And so what we're looking at are the hydrolases. We want something that is going to hydrolyze our ester. Or more specifically, we're looking at esterases is what we want for our job this week. Okay? And so if we look at kind of general information about enzymes, they can be very specific towards compounds, including being specific for one enantiomer versus not working with another enantiomer, depending how they fit into the binding site. Okay? And so depending on the enzyme and what the binding pocket is set up to accompany or to accommodate, it may or may not be able to accommodate the other enantiomer. And a lot of times in living organisms, it's going to want one versus the other. And so there's 3 models we're going to look at for how the enzymes and the substrates come together. Okay? And so the first one is the lock and key model. And so with that model, you've got one lock, one key. So there's not a lot of variation of what is going to work with that enzyme in that binding pocket. You've got the binding pocket is going to be very specific for that one substrate, and it's not really going to accept a lot of other substrates. The second one is the induced fit model. And so that is more like if you think about baseball glove and a ball going into the baseball glove, it's not going to be as specific for one specific ball, but it can fit around a number of substrates. So it will have some specificity in the binding pocket, but it will be more accepting to more than one type of substrate. Okay. And then the third one we're going to look at is called the binding pocket. And so with that one, you've got the binding pocket, and it's got a little bit of variation that is accepted for what will fit in there, but you're going to have kind of the same general structure of substrate is going to be accepted in there, and you're not going to get like huge difference in variation of what will be accepted in the binding pocket. So this is the model that's most accepted for pig liver esterase. So there's been a lot of work as far as enzymology in researching pig liver esterase. We don't know a whole lot about orange peel esterase. And so for this experiment, we're going to go off of the information that we do know about pig liver esterase, and it is expected that the binding pocket model is what fits best with pig liver esterase. So it'll accept our diester compounds in that binding pocket to a certain extent, but if that diester became too large, then it wouldn't fit in the binding pocket as well. So, you know, if we had, you know, 6 carbon chain over here, 7 carbon chain over there, it's probably not going to fit well in that binding pocket. So it is still a little restricted, but will potentially fit the compounds that we've made into that binding pocket. And so what we will be thinking about with our enzyme catalysis versus what we could do, which would be chemical catalysis, is what would be a better possibility. So we're going to look at the mechanism of both acid and base catalysis. For ester hydrolysis. And that's basically what we are looking at with these enzymes as well. So if we look at chemical hydrolysis, we've got two options. We've got acid catalyzed hydrolysis and base catalyzed hydrolysis. And you'll cover these when you cover esters in McMurray. So for the chemical hydrolysis of what I cover today and looking at the mechanism, for McMurray, for the 6th edition, it's on page 793. For acid catalysis, for the 7th edition, it's on page 811. And then for base catalysis, for the 6th edition, it's on page 792. And 7th edition, page before again, it's on page 810. Okay? And so, with both of these, there's not a lot of specificity of what they will react upon. Try to make this a little more visible here. They differ in that one is basic and one is acidic, but they aren't going to be super specific for working on, like, looking at your diester, attacking one side of your diester versus the other. So with the acid catalyzed hydrolysis, we're in the presence of an acid. That's going to help protonate the oxygen of the carbonyl. And so that allows for water to then attack the carbon of the carbonyl. And so then we get this four-membered but charged transition state. So we've got to get rid of that charge. So we then have, since we're an acid, our base is then going to be water, or conjugate base of our acid will then deprotonate the oxygen of the carbonyl. We'll reform that carbonyl, and then we'll lose our alcohol or our ROH group off of that carbon. So in the end, and this works with both acid and base catalysis. Sorry, this is not so clear today here. In the end, you are reforming your carboxylic acid, or you're always forming a carboxylic acid, and then you are also getting an alcohol that is formed, okay, depending on which side is hydrolyzed. So you'll have carboxylic acid and an alcohol, and then in the case of acid catalyzed hydrolysis, you're regenerating your hydronium ion because you've got acid catalyst present. For the base, it's going to be a similar mechanism, but we're now basic. So we're going to attack our carbonyl with the base, and then we reform our carbonyl and lose our R' O group. Okay. So then we've formed our carboxylic acid, and then we need to go through and we'll protonate our alcohol, but then we need to reprotonate our carboxylic acid with an acid workup, okay? And so in the end, the same thing would be formed. We get carboxylic acid that would be formed from that ester that's hydrolyzed, and an alcohol that would be formed from the ester that's hydrolyzed, okay? But again, we're not specific to which ester we're reacting with, or whether it's a methyl ester, ethyl ester, propyl ester, okay? So that's why we want to take a look at enzyme hydrolysis, because we're hoping that we'll have some more specificity. And so this is in your lab manual. And what is accepted for pig liver esterase is this triad. And so this is the serine hydrolase, catalytic triad. And this is the accepted mechanism by which pig liver esterase goes through hydrolysis. And so there are three important residues that are present for the triad. We've got serine, we've got histidine, and then we've got aspartic acid or aspartate, depending on which form it is in. So in the binding pocket, here are our three residues. Aspartate in this case, because we've deprotonated, the histidine, the serine. And so we've got these three guys together. When they come together with as the three residues and then come in contact with the ester, they go through this sequence of using the aspartate to deprotonate the histidine, which then has a movement of electrons through the histidine that then help to deprotonate the serine, which then helps to attack our ester, okay? And so then, in doing that, we now have our ester, or a portion of our ester, attached to the serine, but in the process of this, we also are going to have electrons move up into the carbonyl and back down and lose our OR prime. And so we get formation of the alcohol, okay? But the thing is, we need to regenerate our enzyme. So there's another step to that mechanism. And so we've got to get our triad back. So we're in an enzyme, we've got plenty of water available, so that's going to start the kind of backwards through the triad. We'll use the water to attack the carbonyl that's attached of that ester portion to the serine. Electrons move up, electrons move back down. We'll deprotonate from our histidine this way, move electrons, and move over to our aspartic acid, and then deprotonate that again. So then what we have when we come back, here and here we have what we started with, and out of it we have gained our carboxylic acid, and then we also gained the alcohol, okay? So the same thing happened with chemical hydrolysis. It's just hopefully with the enzyme we're finding some specificity for what we are trying to do and which ester we're trying to attack, okay? So what's important with this, with the triad though, is that it is still, so we can't put those three amino acids together and just get those all joined together and expect the same thing to happen. it does have to happen in that binding pocket. So the binding pocket is important, even though these are the important residues in the whole process. So you can't have this happen without that pig liver esterase binding pocket holding on to the enzyme while these reactions take place. Now, something else that we need to consider through this whole process is how, when we're talking about enzymes and enzymology, how do we keep track of what is going on with the enzyme? And so you'll have an enzyme plus its substrate. They will come together to do whatever the enzyme is going to do with that substrate, and then you should have release of the enzyme and then whatever product was supposed to come out of that enzymatic reaction, okay? And so this whole process is called one turnover when we go from here over to here where we regenerate the enzyme. And so this is important for looking at the efficiency of the enzyme and how well that enzyme is working. So ideally, you get many turnovers for one enzyme working the reaction, okay? And so keep this in mind for that the final report that you're going to work on for experiment 20. You want to consider the efficiency and how well the enzyme turned over and reacted on the substrate. Okay. All right. So in our possibilities of what can happen, we've got a lot of things that can happen. Okay, so we're taking our 20B product and we're going to put it into our enzyme solution, and you're going to follow the instructions in your lab manual for pig liver esterase if you're using that, or orange peel esterase. Okay, but we're taking our diester from part B that has 2 esters on it, and we're going to try and figure out what happens to it while it's in contact with that enzyme. Okay, so one of the possibilities of what could happen is you put part B in, you get part B out. Okay, so basically the enzyme would have had no reaction on its substrate. If you get the diester, start with a diester and get out the diester and nothing happens to it, then the enzyme didn't do anything to that substrate. Okay, and so that is a possibility that you start with B and end up with your part B product. All right, and so as you're watching your reaction, if it doesn't look like that the reaction is progressing, that is okay. For that one data point for the technician number, potentially you may not get any reaction, but we need to have that data point for the whole set of data. Another possibility would be, and this is what we want, is that we chew off the side of the ester that you made 2 weeks ago. So the ester that was made from the acid chloride, we're going to chew off that ester and go back to our part A product. That would be ideal for this situation, okay? And hopefully get it to go to completion as much as possible in the allotted time. Another possibility would be what we call our D possibility, would be to chew off the ester that we made in part A and leave the carboxylic acid, but leave the ester that was made with the acid chloride. Okay? And so far you have data and TLC information and NMR information for B, you have information for A, you don't yet have information for D. Okay? Final possibility would be that it is not specific to either side of the diester and you eat both sides. Okay? And so then you get back the starting material, and when we're talking about starting material, we're talking about starting material for the entire experiment 20. So the experiment 20 started with 4-hydroxybenzoic acid. So that means you would get back 4-hydroxybenzoic acid. Okay? So these are our 4 possibilities of what could happen, and we want to watch out for those 4 possibilities when you're running the reaction. Okay? And so a couple specifics before we look at the data that you're going to put together. So for the reaction, first of all, you want to use at least 0.1 grams of your Part B product. Okay? So hopefully everybody has at least 0.1 grams to use. And remember, you do want a little bit of your Part B product set aside for TLC. Okay, so if you haven't done that, make sure you do that before you start this reaction. If you have more than the 0.1 grams, if you're using pig liver esterase, you can use up to 0.2 grams. Okay, so you can double the recipe. All right. And if you use 0.2 grams, make sure you double the amount of esterase that you're using, as well as we're going to put our diesters in a solution of acetone before you add the enzyme. And so you want to make sure you double your solvent as well. Okay. Orange peel, you're going to use up to 0.3 grams. And again, you would triple the amount of orange peel esterase and triple the amount of acetone that you would use. Okay? So we're going to get these mixtures together for our reaction. And what we need to do is we're going to TLC, we're going to monitor our reaction by TLC. So we're going to TLC first at time equals 0, then at 1.5 hours, then at 3 hours. Okay, so since we're looking at potentially 3-hour reactions, we want to make sure that we have plenty of time for that. So this week, like the first week of experiment 20, you're going to come in and set up your reaction before you go ahead and take the quiz. So we'll get everybody's enzyme reactions set up and everybody's first TLC plate spotted, and then you guys will go on and take the quiz, and we'll give you some other pre-lab instructions. Okay? So plan on coming into lab ready to set up your reaction based on what you have. Okay? Now with the TLC, we are going to, our enzymes are in aqueous media. Okay? So we're going to be spotting something in H2O. Okay? Which we haven't done before. We've only used solvents that evaporate. So you have to be really careful when you're spotting a TLC plate. It's not like the water is going to disappear really quickly. So you've got to carefully spot the TLC plate for your reaction mixture, and then make sure that spot dries before you put more on there. Okay, so you've got to take more care when you're spotting your TLC plate this week than you have TLC plates than you have in the past. You also want to make sure that you're comparing to your 20A and 20B products, okay? So you should have TLC standards for both of those, all right? You're going to use the same eluent, the 95 dichloromethane, 5% methanol that you used a couple weeks ago, all right? But do be careful with the spotting of the TLC plate because you are spotting in water, okay? Now, at the 3 hours, even if your reaction has not gone to completion, you are going to stop at 3 hours. You won't let your reaction go beyond 3 hours. You're going to start the workup. And in your lab manual, it gives you workup instructions for if you have pig liver esterase or if you have orange peel esterase. Okay? So make sure you follow those. The one thing to keep in mind is for orange peel esterase, it sometimes will form emulsions. Not as often with pig liver esterase. So if you need to use sodium chloride solution, that'll be available to use that to help you with the emulsions that may form. The other thing with orange peel esterase, because it may form emulsions, when you put things in the separatory funnel for the workup, don't shake it really crazily because it'll make a good solid emulsion that won't separate out. Okay, so kind of shake and vent nicely, but don't shake it real vigorously because then you'll end up with a problem with emulsion for sure. Okay. Now what you need to do is also be aware of impurities that you may have in your Part B. So from your NMR and IR and TLC data, keep in mind impurities that you had in your Part B, because that's going to probably carry on into your Part C reaction. Okay? So once you are done with the 3 hours, you're going to follow the workup instructions. Everybody will dry with mag sulfate. You'll rotovap. So remember, know your weight of your round bottom flask. Do a good job with this step so that after you rotovap, you don't have visible water spots in your water droplets in your mixture, because we'll send you back, have you take it up in ether, have you do the drying step again, go back and rotovap. You'll use the high vacuum pump again. And then you need a yield. And you need an NMR sample. These are the 2 most important things, okay? So collect the yield, know what that yield is, so know the weight of your round bottom flask before you start rotovapping. And then you're going to prepare your NMR sample with 1 drop of your product in an NMR tube filled to 5 centimeters with the deuterated chloroform. If you still have material left, then you can collect the IR spectrum. Okay, but make sure that you get the yield and the NMR sample first before you go on and collect the IR spectrum. Okay, just in case things are running a little bit low. Now, then what we're going to look at, once everybody does their individual reactions, we're going to put together this table of data for all the 18 possibilities, okay? And so kind of forget the extras down here. This is for a class that's got more than 18 people, but we've got 1 through 18 possibilities. So for your technician number, and different profs have different ways of filling out this table, but somehow you will be filling out this table, okay? You're first going to give the max time, so how long did it stir? That's what max time is, okay? Did it stir for an hour and a half? Did it stir for an hour and 45 minutes by the time you develop the second TLC plate? Did it stir for 3 hours? Then instead of yield, we're not going to calculate a % yield. There's not a real easy way to do that because of all the possibilities of what we could have in there. We're going to calculate a mass recovery. So you're going to take the mass of what you end up with divided by the mass you start with times 100%. Okay? Then you're going to figure out how much of each of these products you have in your mixture. Okay? Now, what you are going to use for figuring these out is your NMR. So can you think of what would be helpful from your NMR for figuring out that information? What was that?
Integrals.
Integrals of what? What specific area are you going to look at in your NMR? Just kind of think about where you've had impurities and other peaks. We want to use something that doesn't have a whole lot of muck in it so we can figure out what's in there. So which area of the NMR will you use? In the aromatic region, right? Because that's where things are clean for the most part, okay? So people with their different ester groups, especially from the acid chlorides, there's some muck in some people's NMR, but for the most part, everybody's NMR should still be clean in the aromatic region, okay? So you're going to look at the aromatic protons and figure out from your NMR how much of that product in the NMR is potentially starting material. So remember, starting material is 4-hydroxybenzoic acid. How much is product A? How much is product B, so no reaction, and how much is product D, okay? And then, depending on your prof, they may have you add additional comments too about your information. So instead of a report form this week, this is what you're going to be reporting to your lab prof into the rest of your class, and this is what we'll be discussing in lab next week during the data discussion, okay? So make sure you do a good job with figuring out what products you got or products you have in your mixture, and remember your TLC and your product mixture should correspond to each other too, right? Okay, questions with that? Okay, I'm going to do quick transition, and we'll look at the practice exam. So let me just get this moved over so you guys can see a little bit better. You guys, if you come up afterwards, just be careful of the cord there. Alrighty, so now we'll go through the practice exam as quick as we can so we can get through the whole thing. And I will post, later this week I will post the key that we're going through on Moodle, okay? So the exam itself is already posted on Moodle. Here's our practice exam. Remember, don't forget, mark whose lab section you're in. That's easy point there, okay? All right, so first series of questions, we are looking at this compound here. We've got methylcinnamate, and it was prepared by Wittig reaction. So we need to know in the first question which pair of starting materials could be used along with appropriate base and solvent to make cis and trans methylcinnamate. And so any ideas on that one? Would this one work? Yeah. Yes? Would this one work? Both of these would work, right? So either A or B is a possibility. Okay, in which of the ways will it be possible to tell apart HA trans and HA trans and HB trans? They'll have a different multiplicity, different splitting patterns. They have different splitting patterns. Same, right? Will they have different coupling constants? Nope, they won't have different coupling constants. How about chemical shift? Yes, they will differ in chemical shift. Okay, how many inequivalent aromatic protons are there in trans-methylcinnamate? So we're looking at the aromatics. How many different? 3, right? Hard for me to cover here. So we got ortho, meta, and para are all going to be chemically different. All right? Sometimes they show up, kind of overlapped on each other, but they are actually chemically different. All right? Okay, the last one. What would happen in your column chromatography separation if instead of using 4:1 hexane ethyl acetate, you use 40:1? Would there be no change? There'd probably be a change, right? The more polar compounds would elute first. So would you flip elution order? No. The less polar compounds would elute first. And last time they eluted last? No. Now all the compounds would elute much sooner? No, right? Because you're going from something that's 4:1 to 40:1 in the hexane, so that means it's less polar. And so E, now all the compounds will elute much later, but in the same order as before. Okay, so we got a lot to read here at this top, so I'll kind of try and go through the highlights here. We had people who were making the methylcinnamate, okay, and different people were doing different things with their column after they made the methylcinnamate. Fractions 2 and 5 and 10 showed UV absorbing components on the grid TLC plate, so they developed those fractions. Student 1 used the rotary evaporator for only fraction 2 and analyzed by NMR found that it was mostly trans-methylcinnamate. Student 2 combined and rotovapped fractions 2 through 4. Okay? So what will student 2 see in their NMR? And they use fractions 2, 3, and 4. So what will you see in the NMR? And this is starting material. See starting material, cis and trans product, right? Okay. What will, which student will have the most material to go on to the next step? Student 2 would have the most material. Which student would have the purest material? Student 1, that owns only the trans-methylcinnamate. Which student had the higher percent yield if you took care of NMR impurities in your NMR and so forth? Student 2 is still going to have the higher yield, right? Because they'll have both the trans and cis product in there. Which student's result more accurately indicates the true cis-trans selectivity? Student 2's. And which is the probable identity of this low spot here in fraction 10? What's that? So you got starting material here of your aldehyde. What would be? Triphenylphosphine oxide. Or what would be something else that would be superpolar? Cinnamic acid. Which is more polar, trans-methylcinnamate or cis-methylcinnamate? Cis is more polar. So you know from up here, this top spot was trans because that's what student 1 isolated. So then the second spot is cis, which means it has a lower RF, so it'll be slightly more polar. All right. Okay, so next set of questions here, we're looking at Fischer esterification using this top set of reactions. Which of the following intermediates or mechanistic steps are not likely to occur? So they won't occur. So which of those possibilities? D, right? All the rest of these would, remember Fischer esterification, acid catalyzed. All of these involve steps that are involved in that process. Step D, we don't have a, we have multiple steps and we have intermediates involved versus having everything occur in one step, okay? In this reaction, the equilibrium is driven to the right by using acid catalysis, excess amount of methanol, heating the reaction at reflux, distilling water from the reaction as it's completed. B, using excess methanol. Okay, which of the following TLCs represents partially completed transformation of benzoic acid to methylbenzoate? Column B represents the halfway point through the reflux. And column A is the reaction at time 0. So which of those TLC plates shows partial product and partial starting material for B? It's going to be C, right? Because benzoic acid is going to be more polar than the benzoate product. Okay, so that's why it's C, not D. All right, so reaction again, top of the page. Student used 3.62 grams of benzoic acid and excess methanol and sulfuric acid is callus. They get 2.42 grams of methylbenzoate. We need to find the percent yield. So first of all, we need to find how much benzoic acid we used. And then so we've got this many moles of benzoic acid and how much of the methylbenzoate we then would have formed. Then take our 2.42 over our theoretical yield. We get a 60% yield. Okay. This question, we've got student loses backpack at Phelps. It's got their organic lab notebook in it, and they had just completed experiment 20B, but they hadn't analyzed their data. They don't know what they made in 20B. But they have their NMR still. They could still access their NMR data to figure out what they made. And so here is the NMR data. And from here, we need to figure out what they made. So what do we have in this region? Aromatics. What type of substitution? Para substitution. Then what do these protons show? What groups do we have present? We've got ethyl group and methyl group, right? Methyl group, singlet, ethyl group here. Okay, which side is the ethyl group on? Is it the part A ester or the part B ester? Part A, right? It's more upfield. And then the methyl group is shifted downfield because it's part of the part B ester. So this is the product that would have formed. Okay? And you would want to draw the structure and also put any information that you use to figure out what the structure is. Okay? So when it says show the work, write out all the information that you figure out. Okay, in 20B, the reaction mixture is washed with HCl and NaOH. Explain the purpose of these washes. Okay, so HCl wash, what is that used for? Gets rid of the pyridine, right? Okay, so you could write that in words, or in this case, you can show the reaction that takes place. Okay, what's the NaOH wash used? Get rid of what starting material? The 20A, right? It deprotonates the 20A at the phenol, so you could write that out so it goes in the aqueous layer, or show the actual reaction. Okay. Student has 20B, or is doing 20B or 20C reaction. They start with 0.27 of their 20B product. They use picoliver esterase. When they're done, they have 0.14 grams of product. What's the mass recovery? So just like we were just talking about, take the 0.14 grams, which is produced, divided by the initial 0.27 grams, get a 52% mass recovery. Okay, let's see if I can do this without showing everything. Okay, we've got these two compounds. We want to know, can you use the DNP test to tell the difference between the two?
Yes. Sure.
Yes. Which one will it react with? It'll react with the aldehyde, but not the ester, so you would get a reaction to form the yellow-orange precipitate with the aldehyde, but not the ester. Okay, we've got NMR here, as well as the data that it was a positive sodium fusion test for this compound. Here is our formula. It also had a positive ferric chloride test. What does that tell you? It has a phenol. Okay, and here is the information for it. So we need to draw the structure. So first of all, what kind of substitution do you have? Aromatic, right? And para substitution. What is this representing? The NaOH, right? Now we have an aromatic ring, but we have a C5. So where do we think the nitrogen is? In the ring itself, right? Okay, so it's para substituted. We had an OH group. It was a phenol. That's the final product. Okay. All right, here we go. 1 minute for the last page. A compound with the following IR spectrum is most likely to give a positive result for which characterization test? C, steric ammonium nitrate, because of the OH group here. Okay, we've got this unknown here. It gives a reddish purple color when treated with hydroxylamine and 5% ferric chloride. So what does that mean? It's an ester, positive test for an ester. We've got this NMR spectrum. What is most consistent with that NMR spectrum? What compound? It's going to be B, right? The ester. And so here we've got monosubstitution. So So we've got our aromatic protons. This methyl group would give this signal here, and then our 2 methylenes would give these 2 signals here. Okay? Questions at all with that? All right, like I said, I will post this on Moodle towards the end of the week so that you do have it to look over again. And I will see you next week for the exam at 4 o'clock.